Thank you very much.
And otherwise, we have Bhavna, as I said, to carry on with the next speaker. Thank you very much, Michael, and thank you, Bhavna.
Please.
Good afternoon, everyone. My name is Bhavna Verma. I'm currently working with Dr. Peter Lawrence at the Department of Zoology, University of Cambridge. I will be chairing the next two talks. Each talk would be for 30 minutes with 5 minutes for discussions at the end. Our next speaker is Dr. Hammed Badmos from the University of Glasgow. He is an important collaborator to DrosAfrica, and today he will be talking about Drosophila, a model for cancer research. To you, Dr. Badmos.
Right. Thank you so much for the invitation to be part of this fantastic program. I've really enjoyed it. And I've really learned a lot, and I can't really wish for more. So thank you so much for organizing this, and to the organizers as well. Right, my name is Hammed. So I'm going to tell you some story about Drosophila in cancer research and how we've used this to actually model some aspects of cancer. Right, so my talk is going to focus on some introduction. I'm going to tell you about why we work with flies and something that you've learned from others in the past, and also some of the tools and technology. I'm going to tell you about cancer in flies and the therapy in Drosophila, and also in personalized medicine. And since I'm going to talk about work that has been done by others, like largely by others, when I say we, I mean they, like people that have done the work. Right, so I started my background or my journey at the University of Ilorin in Nigeria. So I did my degree there, starting as a 16-year-old, which was really unusual for me then, and I had to learn my way through the university. But it was a very good experience because I was really lucky to meet a lot of mentors there. And then migrated to the University of Liverpool to do my postgrad and PhD. And presently I'm at the University of Glasgow, researching at the Wolfson Wall Cancer Research Center. So over the years, like I said, I've really mingled with a lot of mentors, and actually they've really inspired myself and my science, and continue to do so. So to foster the use of Drosophila in Nigeria, so I'm part of the pioneer of the Drosophila Nigeria, and thanks to Rashida for really pioneering this and also leading the team. So I'm also part of the GLTC as a collaborator, and thanks to Dr. Abolaji for actually promoting this and actually leading the effort. So I'm going to throw you to the floor that, these two organizations actually need your help. So if you can help them financially, that would be really helpful. And I can tell you that they're going to use the money properly. Right, that's the introduction on the general one. So now focusing on my talk for today. So the big question that we're actually trying to tackle is, how do cells continue to proliferate and then invade or break the basement membrane to migrate to distant sites? And scientists have used several models to do this, including mouse model, including zebrafish, including in vitro system, organoid 2D cells, the related system, organ on chip system, and we are using Drosophila to model this aspect of cancer. And the reason why we use Drosophila is because there's a lot of conservation between Drosophila and human. So we've got this digestive system that is conserved, the genes, the cells there are conserved, can be more complex in human, but we can actually model some aspect of it in Drosophila. We can also look at the nervous system. Several speakers have shown you the power of actually looking at the Drosophila brain and how you can use that to model locomotion, aggression, and everything related to human. Also, the power of fly is that we can actually dissect out different tissues. So we can actually look at them not only at the cellular level, but also at the tissue level. So you can begin to ask, okay, what is really required for the organization of this particular tissue, the antenna or the eye? And you can begin to ask, is that gene probably also required for other part of the system or other part of the organism? So you can actually go to the cellular level. And as Eric mentioned yesterday, Drosophila continues to allow us to actually go to the roots of any problem that we want to solve. So Drosophila is really good for those type of difficult questions. So also we can also look at Drosophila from at early stage, from the embryo stage to the larvae stage, until the pupa stage, and you can begin to ask, okay, I'm looking at the gut. So what is that gene that is actually required for the development of the gut? What is that gene that is actually required for development of the gut in the embryo, in larvae, and in adult? Is that conserved? What if we remove something in the embryo? Can we see that in adults? So you can actually look at the stage, because people have shown that the life cycle is actually really short. So those are the power of Drosophila. So I'm gonna tell you about some of the tools that we use, and the most popular tool is actually the GAL4 system. And the power of this tool is because we can actually drive the expression of any gene. So we can knock down genes, we can overexpress them in a tissue-specific manner. For example, assuming you've got a fly that carries what we call the GAL4, and then you've got a fly that also carries the UAS, that carries the UAS construct. So we can actually cross these two flies together, and in the progeny, you can begin to generate flies that expresses your gene of interest. For example, here we're looking at green fluorescence protein, GFP, in your wing. So that is really the power of this type of system. Another system that we've got is the LEX-OP system, which is also a binary system and allow us to also express gene in a similar manner to the GAL4 system. So another system is the QF-QUS system as well, a binary system that also allows us to interrogate genes similar to the GAL4 system. And these several systems are actually powerful because you can begin to look at cell-to-cell communication, cell-to-cell interaction, and you can begin to ask, okay, if I knock down a particular gene with the GAL4 system in the wing, can I knock down another gene in the head? So these three or two independent systems allow you to do something like that. And for our case, we can begin to look at organ-to-organ communication. For example, the effect of the cancer microenvironment on cancer. We can begin to look at the tumor microenvironment on tumorigenesis as well. So these are the powerful tools that we've got in the fly world. So another recent tool that we've got is the CRIMIC technology, and actually it's powerful because you can actually insert a disruption into the genome of the fly, and then you can insert this at a particular gene, and then that leads to the disruption of gene expression. It is of interest that you can flip out, so you can remove this insertion in the fly, and then you can generate a full gene transcription. And also you can swap this cassette. So it's a cassette that insert, you can also swap it with other cassettes. So for example, assuming you inserted a GAL4 into the genome, you can swap it with GFP, and then you can look at the localization of the particular gene that you're interested in. You can swap this with LAGZID and look at the expression of the particular gene that you're interested in. So all the systems are actually in vivo. So another thing is that you can actually rescue your gene of interest. So for example, you've disrupted a particular gene function and you want to ask if what I've disrupted is actually right. So you can overexpress the cDNA of this particular gene, and then you can rescue this phenotype. And this is better than using RNAi construct because you don't care about off target, and actually you know what you're dealing with in the system. So having shown you the technology in flies, right, so I'm going to show you the journey of Drosophila in cancer research, and actually how Drosophila has really influenced the cancer community, and how the cancer community has also influenced Drosophila research. So I'm going to talk about some of the important figures in this journey over a century. So the first person to actually show that flies can develop tumor is Mary Stark, and she showed this about 100 years ago. And about 60 years ago, Elizabeth Gerde was the first person to actually show that there are tumor suppressors in fly, and was the first set. So I and my colleagues were the first people to actually isolate the tumor suppressors in fly, including Scribble, LGL, and others. And about 20 years ago, Elena Richardson and Tanshu were the first set of people to actually look at cooperativity or activation of oncogenes and loss of tumor suppressor in fly. And Ross Kagan was the first guy to actually cure flies of cancer. So these are people that have actually contributed to the fly world over the years in cancer research, and and they continue to contribute to it as well. And we're not really bothered about just curing flies of cancer, but also looking at how can we actually cure patients. So that's actually the overall goal in this field. So I'm going to show you about some of the models that we've got in cancer research, and just some Drosophila model that are actually used to model cancer. So one of them that I've personally worked with is the Drosophila egg. So, and the Drosophila egg is found in the ovaries of flies. So there are 2 ovaries in fly. So the ovaries are made up of ovarioles, and the ovarioles are actually strands of egg. And the egg can develop from the germarium to stage 14. And actually we can image all the process of this egg development. And there were 2 cell types in this system, so in the organ. So we've got the somatic cell that actually surround what we call the germ cells. And this somatic cell actually forms the epithelium of the egg. So basically we can look at epithelial cancer in this type of system. Another power of Drosophila egg is actually we can image this thing over time. So we can begin to look at every stage of egg development over a long period of time, and for as long as actually 10 hours. And for example, here looking at stage 5, we can see the egg rotating. And actually that has been shown recently, actually last week, in organoid model, that actually doing morphogenesis of organoid, there is also a form of rotation in this organoid. And this was shown in Drosophila about 20 years ago. So showing you how powerful Drosophila can be in modeling some aspect of things that we cannot actually model in patient or in human. So, and also can also allow to probably ask and answer some questions. So for example, you can ask, can you stop this rotation? What are the genes that are required for this rotation? So these are the things you can ask in Drosophila. Another stage is the stage 8 to 10, where you can see cell migrating towards the posterior end of the egg, and the migrating in synonymous to what we can see in cancer cell, because cancer cells tend to invade their environment. So we can begin to ask, what are the genes that are required for this migration? Can we stop this migration? Can we actually drug this migration? So these are things you can ask. And the beauty of Drosophila egg is also you can actually image them for a long time, and you can begin to look at not only egg development, but also the early stage of embryogenesis. So that actually gives us the power in Drosophila egg, which in other tissue can be difficult. So using the power of this tool or of this imaging and also the Drosophila egg, we can model some aspect of cancer. Namely, we can model tumorigenesis, and also we can model invasion. So for example, the somatic egg, the somatic epithelium is actually covered by a layer of somatic cell. But when we remove a particular gene known as ataxin 7, we saw not only multilayering, but actually invasion into the oocyte. So clearly we can actually model some aspect of tumorigenesis in this system. Another system that I'm going to show you is looking at the Drosophila eye disc. So we generated tumor in the eye and labeled with green fluorescence protein GFP. So in wild type, you can see localization of, so this is the O larvae. So you can see the localization of the GFP to the eye disc, and when we dissect the tissue looks normal. When we activate a form of RAS known as oncogenic RAS, we saw also a bit of overproliferation of this eye disc, and that's the localization to the eye disc in the O larvae. And then we can look at the dissected tissue as well and saw a bit of overgrowth. So when we remove a tumor suppressor known as Fribo, we saw a reduction in tumor, and also this is the dissected tissue. But the striking thing is when we combine the loss of tumor suppressor with activation of oncogene, we saw massive overgrowth of this eye disc and of this eye disc or the cephalic complex. And actually you can see the dissected tissue. So clearly we've actually modeled some aspect of cancer in this system. And the next question is, can you actually stop this? What can you use to stop this? Can you do this genetically? We can do that in flies. Can you do this using drug screening? That's what we can do in fly. So these are the beauty of using Drosophila for cancer research. So another powerful system is actually tumor transplantation in fly. So Tana mentioned a lot of what is done about this, and it actually is pioneered the field very well. So you can actually inject some cancer cell that you know that they're actually cancer cell from fly. You can actually inject them into fly, and normally when you inject normal cell, they won't grow. But when you inject cancer cells into fly, you can see this massive overgrowth, and not only massive overgrowth, but also they tend to metastasize. And Tani showed yesterday about how he has used some suppressors to actually suppress this growth. And another thing you can also do that is doable in fly is actually suppressing gene in the host. So you can suppress gene in the tumor and also suppress gene in the host, and actually ask, can this prevent this overgrowth? Can this prevent the metastasis? So these are the powerful things we can do with flies. Another system that we've looked at is looking at nutrition and tumor. So we looked at the wing disc is a powerful epithelial cell, and Paul showed yesterday about what he has done with cell competition as well. So we use this a lot in Drosophila world. So what we looked at here is a gene known as EGFR. So EGFR is a receptor that is also involved in several cancers. And what we found is that when you activate EGFR in this wing disc, you can see a form of hypoplasia. But when you feed the fly with high sugar diet, the wing disc is normal. So this is a normal morphology of the wing disc. But actually, when you combine high sugar diet with activation of EGFR, that led to neoplasia. And that is really phenocoped by actually overexpressing a glucose transporter known as GLUT1. And it is of interest that when you knock down lactate dehydrogenase, you can actually reverse this neoplasia to hyperplasia, showing the dependency of cancer cells to what is called the Warburg effect. And it is of interest that we can actually do this. We can model all these aspects in Drosophila, not only for sugar, but you can also ask what is the influence of lipid. And Carlos mentioned yesterday about how he has got this chemically divine diet, and you can actually use this power to actually model anything you want to model in relation to nutrition in Drosophila. Another model that, so I've shown you several models, single eat model about things that are probably not required in human cancers, but we've modeled an aspect of human cancer known as a colorectal cancer model. And actually, we've picked two drivers of this colorectal cancer and the reoccurring mutation in patients. One is, so we looked at the Drosophila gut because actually that mimics what
in the plasma reticulum where it undergo modifications.
Right.
Yes, those are the basics you understand from the cell. You don't have problem with one. Yes.
Sorry, yeah. Farida, do you want to mute yourself?
So from the cell.
Farida, do you want to mute yourself, please? Thank you.
I've muted her now. I couldn't find her. Yeah, it's done.
Cool. Thank you so much. Right. So we're trying to model colorectal cancer, and this is a cancer of the colon, right? So we're using the Drosophila gut, so showing the mimicking of this type of cancer. So when we looked at wild type clones, so we generated clones and marked them with GFP here. So looking at wild type clones, you can see individual clones both across the gut, and they don't proliferate. And also you can see similar thing after 4 weeks in the control gut. So when we look at loss of APC, so loss of APC occurs a lot in colorectal cancer. So we saw a bit of proliferation, and we can see this proliferation compared to the wild type. When we looked at activation of oncogenic RAS, we saw loss of this RAS, and which is really different from what we saw in the eye disc. So, and when we combine loss of APC and activated RAS, after 4 weeks, we can see this tumor growing. And it is of interest that when you actually pump up this level, you can actually see metastasis over time. So, and here I've shown you how we model using two drivers of colorectal cancer. We call this a 2-hit model. But actually in our lab, in the Kagan lab, we can actually model several tumor model or several hits of tumor models, and we can go as high as possible. And the power of this is that you can use this for several things. You can begin to ask questions, what is the effect of diet on this? Can I actually reverse this? Can I actually treat this? So these are the questions you can begin to ask with this type of model. Another thing that I'm going to show you, so I've shown you several of the models, so I'm going to tell you about how we've actually used this to actually bring therapy to patient. And I'm going to be showing you the Drosophila eye. So here we model a cancer known as medullary thyroid cancer. So it's a cancer of the thyroid, and it is, as at then, it hasn't got any treatment. Is that okay? So here, Drosophila was really powerful in bringing some of the therapy to market. So in conjunction with other systems. So because when you're trying to actually develop drugs, you need multiple systems. So Drosophila was powerful in actually playing its own role. So here we looked at the Drosophila eye. So the normal morphology of the Drosophila eye, you can see the regular patterning of the eye. But when we overexpress a form of red, that is actually one of the driver of medullary thyroid cancer. So we saw rough eye phenotype. So you can see the eye actually growing out of the animal. And it is of interest that we can actually treat this rough eye phenotype by a compound known as ZD6474. And this compound actually led to the first treatment of medullary thyroid cancer, showing you how powerful fly can be in actually bringing drug from the lab to actually to the patient. So another thing we've actually done is actually look at the combination, because we've got this powerful genetics in fly. So we can actually combine the genetics of fly with drug screening or compound screening. So here we looked at medullary thyroid cancer again, and then we looked at the Drosophila wing disc. We looked at Drosophila wing. So the Drosophila wing has got normal venation. But when we looked at an oncogenic form of red, we can see this multiple venation. And it is of interest that when you actually remove a copy of a protein or of a gene known as ELK, you can actually reverse this multiple venation. And you can actually treat this multiple venation as well with AD58. The striking power of it is that when you combine the genetics with a compound, that actually reverses the wing to normal. So showing you how powerful Drosophila can be in actually screening for genetics individually on its own, and the power of actually combining this with drug screening. And this can only be done in a system like Drosophila.
Hamid, we have 5 minutes left.
Okay, I'm almost done. Okay. Right, so another thing that I'm going to show you is actually looking at personalized medicine. And this is a study that have actually been powerful and actually first of its kind in Drosophila world. So here we looked at colorectal cancer patients. So what we've done is that you can analyze the drivers of this patient and the driver genes of this patient, and actually create fly avatar for the patient, and then you can do drug screening and then treat the patient with that. So you're not taking this to the patient directly, but actually the clinician of the patient needs to agree to that. So that is the power of Drosophila. So we've used that for colorectal cancer, we've used that for adenocystic carcinoma, and using the same approach. And the system has actually generated a lot of headlines, and this is one that was written in July. So the idea is actually, though we can use fly to actually treat patients, but because it's actually very fast, we can do this in an old animal system. But also we can also combine this with other systems. So you can combine this with probably organoid, with some mice model, but actually Drosophila is a very good starting point in actually bringing therapy to patients. So altogether, the take home is that I've shown you how we've modeled an aspect of tumorigenesis, proliferation, and metastasis in Drosophila, and showing how Drosophila can be powerful for this. So in future, we would love to actually model tumor heterogeneity and the effect on drug response. It would be cool to also model tumor microenvironment and drug response. And also, we are looking at how to develop better drugs in combination with patient nutrition. So if you've enjoyed what I've talked about all through, so these are people that actually work on Drosophila directly or indirectly. So, and you can drop me a line. I'm happy to share them with you. And they've actually got fantastic models in the labs. So to young folks like myself, so science is an exploration of yourself and nature. So find people that can actually bring out the best in you, because you shouldn't allow anyone to kill your creativity and your curiosity, because you've got a place in world and in science. Thank you so much for listening.
Thank you very much for the insightful talk. We have a few questions. The first one is from Oibo. What is the difference between the migration seen in the stage 8 and differentiation? Any similarity?
Differentiation of what? Sorry, differentiation of what?
Oibo, can you please, would you like to unmute yourself and ask the question, please?
Okay, thank you. I hope you can hear me.
Clearly, please. Yeah.
Okay. I was asking, what is the difference between the migration seen in the cell? I hope those cells were normal cells, egg, in the egg of the Drosophila, stage 8, and when or does differentiation occur also during the embryogenesis stage of the Drosophila?
Okay,
or what is the difference? Yeah.
Okay, very good. Yeah. So these cell types, they're normal cell. So they're migrating. So this is a normal developmental process. So they're migrating actually to, so this is actually a type of video. So they're migrating to that posterior end, and that's the anterior border of the oocyte, to actually go into the oocyte, and they're required for the patterning of the fly. Is that okay? So, and so they need to migrate normally, and in embryogenesis as well, that's normal. So the EMT, so that is also a form of migration. Is that okay? But that is a different cell type. So this cell type, we call them the border cell. Is that okay? And in embryogenesis, that is a different type of cell. Makes sense?
Thank you. The next question is by Yahatu. The question is, what instrument can we use to assay apoptosis in Drosophila?
So the simplest one is you can stain for caspase-3. So you can just stain for caspase-3, and then you can look at apoptosis.
Okay.
Yep.
And I have a question myself.
Yeah.
Have you compared the tumors that you see in fly with what we know in what we've seen in humans? Like, have you seen angiogenesis in the tumors that you observe in flies?
Very good. So remember that for example, the oxygen transfer in flies is different from patient, right? So we cannot model that to a large extent, but to some extent we can still model that, because Drosophila has got a trachea system. So, but really different from the blood flow that we've got in patient. So we cannot model that to a large aspect, if that makes sense. But we can model, you can look at apoptosis, you can look at EMT, you can look at proliferation. So you can actually, and then you can stop these aspects of cancer. So that's what we can